Nothing, dear. Nothing. But I do have. Did you have something?
So, we have a lot to do today, so there's not much time
Why I
to
have
be inefficient. I'd like you to look at your mixed culture. What you should see is the streak might be getting lighter as you go through. If it doesn't, that doesn't mean you didn't do it correctly. It just means the bacteria grew really fast. So what you're looking for is single colonies. Purification, one of them. Here's the other one. On this one, all I see is the big pink colonies. What species of those three pink colonies? Yeah, Espersescens. Espersescens, when we start, so imagine a race, and we set up E. coli, V. subtilis, Espersescens, all of the starting line, and we put it in the end. Espersescens just outperforms the other two species, so don't be alarmed if you only see the pink colonies and you don't see the white E. coli. On one of them, I did get some of the E. coli colonies coming through. And I have 2 colonies of E. coli, but the rest have that pink pigment, which is Serratia marcescens. So sometimes it does catch up, not always. So I'm going to come around and just look at your streak plates, give you a little tip if you need it, let you see my example. I'll put one up here, the other I'm going to carry around with me. You're going to be doing a lot of these today. And I'm going to show you an alternative technique that's just a little faster than the quadrant streak. But I do have to announce something again. So whenever there is anything chemical or biological out on the bench, you need to be wearing your goggles. It doesn't matter if you are doing the experiment or a partner is doing the experiment or I am doing an experiment. So I was notified by our environmental health and safety person that these goggles that I'm wearing today are not going to be sufficient going forward. So I'm waiting to hear guidance on what type of goggles we need, but I know the chemistry style goggles that many of you are wearing that seal all around do fit that requirement. So stay tuned for an announcement from me on whether you might need new goggles. So if you're wearing these type, you might need to just get a pair of the fully enclosed ones. So they do sell ones on like Amazon and some kinds of hardware stores that are big enough, but I do know that they're not going to make any exceptions for glasses and things like that. So you need to find a pair of them, unfortunately. Yeah. So.
Other question.
Yeah.
The standard is to make sure that the mistakes inside the eye.
Yeah, they want them fully sealed.
Okay.
So in the future, Celeste Cregan, Miss Cregan is upstairs on the 3rd floor, all the way at the end of the hall. On the left, in the future, is goggles she has some to provide for you. Today we're okay, but just stay tuned for that announcement moving forward. And listen. So I'm going to walk around and have a look. Good. Oh, you got some E. coli too.
I think it's perfect. Thank you.
Yeah, I do see some E. coli outside the door. So those white colonies are E. coli. I don't see Justin, your colonies are a little crowded, so just make sure when you go from 3 to 4, maybe take a little more from 3.
Yeah,
to
I
bring
think I
it to 4.
remember I was peeking from like this corner.
Yeah,
and
so
then
you
not that
just
far.
missed it. Yeah. So maybe next time, just take it. Okay. So,
Nothing in.
well, you went right from 1 to single colonies. So what probably happened is you
Yeah.
just took very little from one, like you probably barely touched
Oh.
one. So next time, just go
Yeah.
like deeper in. Like don't be afraid of it.
And I'm having a question.
Yeah.
So this is like a smooth, and what does a rough and fuzzy look like?
Rough and fuzzy will look like almost like velvet. So like not shiny. See how like the colonies look kind of smooth in the light and glossy? A rough colony won't look glossy, it'll look more matte. I would make the opposite comment to you, try to make it feeling really happy with your single colonies. You have one single colony. Okay, good. I see some nice examples here. We're going to be doing a whole bunch of experiments with selective and differential media today. So you'll get, if you're not confident in it, you'll get plenty of time to practice today with your colony purification. Okay. So, today we have a whole bunch of stuff. This lab goes very nicely with what we're starting in lecture this week. Sometimes we don't sync up well, but today we sync up. So, lecture material 3 deals with 3 main things. Bacterial population growth and how to determine it. So we don't determine bacterial growth by the size of an individual cell. We determine it by the number of cells. So today we're going to do a technique called the viable plate count, where we determine how many bacteria are in a sample, and thus determine how much we have culture here that I'll make here for us to use. Then we are going to work on selective and differential media, which is another topic we cover in this week. So population growth, how we count population growth, and these different types of media. So over the last 2 weeks, we learned that differential staining allows us to tell the difference between 2 species based on some sort of structural property. Differential media lets us tell the difference between 2 species of bacteria based on metabolic properties, and sometimes cell wall properties, but I'll get to that. Then, we're going to start our Winogradsky setup. I have the report template with me. I'll also be putting it on Brightspace. The Winogradsky column is an enrichment media setup where we're going to look at soil from the Kingsborough farm. So I have some nice soil. We found 4 earthworms in it yesterday. So maybe you'll get lucky and see some earthworms today. We want to pull those out, but we don't want them in the column. So we set up the columns. Yesterday they look pretty good. You'll be setting them up today. We're going to set up 4 columns through the lab. You're going to do this with your lab group. So this is a group report. The group needs to grow. So this is lab group 1, lab group 2, lab group 3, and lab group 4. So we're full section. Now, over the next 6 to 8 weeks, you are going to be coming in every day and taking a picture of all 4 Winogradsky columns, front and back. Not the ones from yesterday's class, but the ones your class will set up today. You're also going to be looking at these control columns. These control columns are columns we set up last year. So Bio 50 students just like you, these were their columns last year. And they're a little further along in their development, so you can make a comparison about what your column might look like in the future based on how these are. What you might see, different layers. Green layer up here, red layer, and then those layers correspond to different species of bacteria that might be living in the soil. I'm going to put up a whole bunch of resources for you. I think I actually have already in the week 3 lab folder that teach you all about the Winograd comb. And you can use those resources to both label your columns and write your report. Every week, you're going to be taking those pictures. Each week, I want you to label those pictures with what you might see developing. Now, not all layers are going to be present in all columns. That's normal. We don't know. This is research. We don't know what's in that soil. That's why we're doing this, to find out. So, this is a group report. If you feel very strongly about working alone, I'm going to say a couple of things. Number one, I highly discourage it. It is a lot of work, this report. It is almost too much work for one person to do on their own well. Number two, I understand group work is difficult. You have to deal with different personalities, different schedules, all that. I will tell you this, all science is collaborative. I have never worked on a project in my entire career completely by myself. I have collaborated with people I've never even met on the other side of the world before Zoom existed. You make phone calls, send emails. Every group member is expected to contribute. Every group member is expected to be given the opportunity to contribute something to their strength. I do not grade these harshly. It's a 75-point report. It's not worth our stress or giving ourselves ulcers. But it is good practice in scientific collaboration and working in a team, because when you get out into a job, you don't always get to choose your team. You kind of chose your team by where you're sitting. But I understand group dynamics can be difficult. So I'm going to say, if you run into trouble with your group, if you come to me 2 weeks before the report is due at the end of May, there is very little I can do to help you. However, the earlier you come to me if there's a problem, and I don't like we're all professionals here. We're training to be professionals, so we're going to act like professionals here. But if there are big problems, you can come to me and we'll try and mediate our way through it and come up with a solution that works for everyone. But what you should do in the beginning is talk to your group members about how you plan on communicating, how often you plan on communicating, and how you expect to do the work on the report. You guys are lucky. You live in an age of Zoom and Google Drive, and where you can collaborate from afar and add things to a document from afar. So, maybe one person's responsible for taking the pictures each week, and then, everyone put in their labels by next Friday and keep that going. Because I do promise you this, if you take all your pictures and wait until you're writing the report to label your pictures, you're going to be very angry at yourselves. Because as we know, sometimes when we take a picture, the colors don't show up as well as they did in person. So what I recommend when you take the pictures, maybe use your edit tool on your phone and just draw in the layers right then and there. Saying, I see a layer here. For the first few weeks, you're probably going to see nothing. It takes time to grow. Don't be alarmed by that. But we're going to set this up today. And again, it's a group report, so make sure you have contact information for everyone in your row. And make a plan to work together. The sooner you do that, the sooner you set those expectations for your group. I have the expectation that you'll treat each other according to the course culture that I set out in the course. It's in the syllabus. We treat each other with respect. We treat each other with professionalism. We're forgiving of one another. And we assume the best intentions. And again, if you have a problem, the sooner you come to me, the better. We'll be doing a second group report, which is the bioremediation report, which we're going to set up in about a week or two. That is, we're going to simulate some pollution in tiny containers in the lab, and you're going to use microbes to clean them up. You're going to take pH data and some microscopic data looking at how the bacteria degrade pollution. And then I'm going to ask you to be a little creative with your group. You can write a news article about the environmental disaster at Kingsborough in the lab and what happened and how you helped clean it up. You can make an Instagram-style video, not a TikTok, they're too short. But you can make, a podcast, a video, anything you like. You can even write a report if you really, really want to, as long as it has the right information that I'm asking for. But you can get creative with it. And that's your second report. That is a 75-point report as well. Then you have your unknown identification. That is your third lab report. That's actually the one that's due first. So I went backwards here. That is an individual report. So that one we will do on our own. Any questions about those? Okay. So we're going to start with today's activities. The first thing, we're going to go in order today. So the first thing we're going to do is the viable plate count. The viable plate count answers the question, how many bacteria are in this tube? And the answer, a lot, is insufficient for this. We want to have a better quantitative approximation. And this technique is used to count the number of living cells that are in this tube right now. Now, over the last 2 weeks, you've heard me repeat over and over, don't worry about getting enough bacteria. One drop contains millions, perhaps billions of bacteria. If I were to take those millions of bacteria and spread them on a plate, we know what happens. That. We can't distinguish the top of your streak, if you can't see what I pointed to there. That. We can't see individual colonies there. What does one colony represent?
Population.
What?
Microorganism.
How many microorganisms?
One species.
One species, but the descendants of one bacteria. So by counting the colonies in the viable plate count, we can extrapolate back and calculate back how many bacteria are in that tube based on how many colonies appear on the plate. And I'll show you how we do that. So what
Question.
yeah.
I should ask earlier, within the platform stage, right? Is this stage or?
I call that a plate.
Okay, so the
Petri
culture,
dish.
Petri dish, culture.
Culture.
In the standard like when you're working, if I don't label it, that we consider accurate information because label the quadrants. If I just
No, as you get better at it, you don't need to label the quadrants anymore. When I do it, I don't label the quadrants, but I always label the plate.
okay, so
I always label the plate with my initials, the date, the species, at minimum. Today, you're also going to label it with the type of media you're using, because we're going to use different media. But the viable plate count involves dilution, but we can't use a streak plate to determine the number of colonies we had in the original culture because we didn't do a precise dilution. I don't know how dilute this is compared to this. So I can't calculate that. I can't say there are 10 colonies here, therefore there are 10 million in the original sample because I wasn't methodical about my dilutions. The viable plate count counts what we call CFUs per milliliter, or colony forming units. And remember, that equals one bacterial cell or a clump of bacteria. So this is an approximation. It's not the exact quantitative count. It's an approximation. In your reading this week, for week 3 annotations and the textbook readings, you'll see that we call this an indirect method of counting. Meaning we're not directly counting the number of cells by going 1, 2, 3, 4. We're looking at some representative of growth. In this case, we're looking at how well it grows on a Petri dish. We only care about the living cells in this, the viable plate count. The viable plate counts are used throughout all sorts of purposes. If you're testing the safety of a water sample to drink, you're going to look to see how many bacteria are alive in it. Dead bacteria don't really make us sick. You might use this in a clinic if you have a patient give a urine sample and you want to know if the patient has a urinary tract infection. So you're going to look for the number of bacteria in the urine. If you work in a food manufacturing facility, you might take a sample of that food and do a viable plate count to see if it's contaminated with an undesirable organism. And if it meets a certain threshold, that might trigger a food recall. We hear about those all the time. What happens is, a friend of mine was a chemical engineer and she worked at a yogurt factory up in Massachusetts, and her job was to take the sample and hand it off to a microbiologist that worked there for testing to make sure that only the lactobacilli that are supposed to be in the yogurt are there and not too many other types are there. So this is about food safety, this is about clinical diagnoses, this is about water safety and environmental microbiology. So this is a very widely used term. So we're going to test for the concentration of bacteria by looking for the CFUs per mil. And again, like I said, if I take one drop of this and put it on a plate, the colonies are going to grow together, so I can't determine. I can't count that. I can't pick out individual colonies or clumps of colonies. So I need to dilute this. But I'm not going to dilute it by streaking like I do in colony purification. Instead, I'm going to dilute it by making serial dilutions in a mathematically set way. That way, when I count my plate next week to see the number of colonies, I can calculate back and determine how many bacteria are in the actual culture. Now, there's a couple of vocabulary terms you need to know with this. We've determined over guidelines and research over many, many years that humans can reliably count between 30 and 300 colony-forming units on a plate, on a petri dish. Anything less than this, and I run into something called sampling error, where I might not just taken up enough of the sample, or the sample might not have been mixed well, and I don't consider that an accurate representation of the culture dilution. If it's less than 30 colony-forming units on the plate, I will call that TFTC, or too few to count. The upper limit of what humans can count on a plate reliably and successfully, because then the colonies start to grow together, is 300 CFUs, colony-forming units. Anything more than 300 CFUs, I'm going to call TMTC, too many to count, or TNTC, if you want to get fancy, too numerous to count. So the problem is, I don't know how many bacteria are in this tube. I want to find that out. So I don't know how much I need to dilute it to get between that 30 and 300. So when we do this procedure, and you're going to do it in pairs, or you can do it in 3 because there are 3 today. You are going to plate several different dilutions and hope one of them or 2 of them fall within that 30 to 300 range. And we have 2 organisms. We have Micrococcus luteus. There are 2 separate techniques I'm going to show you, the spread plate and the pour plate. And then we have an E. coli culture that we're also going to dilute and plate via either the spread plate or the pour plate method. So you're going to learn both. To make the dilutions, we're going to use a technique called serial dilution. That's when you make a dilution, and then make a dilution of that dilution, and then make a dilution of that dilution again. So each group is going to get a tube with a culture in it. And let's see how many we have here. 1, 2, 3, 4, 5, 6 groups. 1, 2, 3, 4, 5, 6 sets of bottles. Perfect. 3 groups are going to do Micrococcus luteus, and these are the dilutions for Micrococcus luteus. We're going to use different dilutions for E. coli, and I'll show you those next. But you're all going to start out by making your dilutions the same way. So, you 2 will do Micrococcus luteus, E. coli. Micrococcus luteus, this whole row. Lydia and Kevin, E. coli. At Micrococcus luteus, E. coli, Micrococcus luteus. So you're going to take your culture tube. Oh. Maybe. One thing always slips underneath my notice. It's gonna fill them off for us. Okay, so we want to know how many bacteria, how many CFUs per milliliter are in this tube. And if you want to role play to make it more interesting for yourself, pretend it's a meat sample from a factory, or a water sample from outside, or a urine sample from a patient who you want to see if they have UTI. Your choice. Sometimes that makes things more fun. You don't have to, though. You could also just pretend this is allowed. So, start out with not knowing how much is in here. So each group is going to take 3 sterile bottles of water. And if you haven't already, please heat up your sterilizer and get it going. I'm going to regret drawing these up so high, but okay. So each bottle contains 99 milliliters of water that's been sterilized. So there shouldn't be anything in them. That is our diluent. The thing we're going to dilute the bacteria into. Then, for our first dilution, we're going to take 1 milliliter out of our culture tube and put it in the first bottle with 99 milliliters of water. 1 part bacteria and 99 parts water gives you 100 parts total. Can we all agree on that 99 plus 1 is 100? I'm taking it really slow. This is the hardest math we do in this class. It's all just multiplying and dividing by 10. So, this is a 1 to 100 dilution. 1 part out of 100 total parts is bacteria. 1/100 can also be written as 10 to the minus 2. So you're going to take 1 ml out of the culture tube and put it in the first bottle, which I've labeled 10 to the minus 2 for me. You have to label the bottles yourself. Put the bacteria in, give it a little mix. These caps are not watertight, so we're not going to shake it like a drink. We're just going to gently mix. Then, we're going to take 1 milliliter of our 10 to the minus 2 dilution, and we're going to put it in the next bottle of 99 milliliters. And this makes a 1 to 100 of a 1 to 100 dilution. And the way I find this easiest to write when you get into larger dilutions, I made a 1 to 100. 100 has 2 zeros of a 1 to 100, 2 more zeros. That gives me a 1 to 10,000. So it's a 1 to 100 of a 1 to 100. So that is 10 to the minus 1, 2, 3, 4. So 10 to the minus 4 dilution. Then mix it up gently. Take 1 ml out and put it in a third bottle. That also contains 99 milliliters of water. So now I'm making a 1 to 100 of a 1 to 100 of a 1 to 100. That's 1 to 100 of a 1 to 100 of a 1 to 100. That's 10 to the minus what? How many zeros are there?
6.
6. Yeah, 10 to the minus 6. 1, 2, 3, 4, 5, 6. That's a 1 in a million dilution. So this bottle has 1,000,000 times less bacteria than our original culture. We diluted it in a very precise and methodical way. Now, for the M. luteus, we're going to plate different dilutions. We're going to plate a 10 to the minus 5th and a 10 to the minus 7th on the spread plate. Can we make a 10 to the minus 5th and a 10 to the minus 7th dilution? No. So how are we going to do that? Okay. So remember, this is CFU per mil, and each time I took out 1 milliliter. So if I take out 1 milliliter of the culture solution and I put it on a petri dish, that keeps the name of the dilution. It keeps that at dilution level because I'm working with milliliter units. So that plate would get labeled 10 to the minus 4. However, 10 to the minus 5th is a tenth of that solution. So tenfold less. You go 1 decimal place, that's tenfold less, right? When I move 1 decimal place, that's tenfold less. So if I take 1/10 of a milliliter out of that bottle, that then becomes my 10 to the minus 5th solution. And that's what I'm going to put on my spread plate for M. luteus. For the 10 to the minus 7, I have 1 ml. That would be a 10 to the minus 6th plate. If I take a tenth of that, 0.1 ml, what is that solution? If I take a tenth of the 10 to the minus 6, what do I label that plate? Large. What is it? 10 to the minus 7, correct. Because it's a tenth less. So we'd add effectively 1 more zero instead of 2. Because we have been diluting 1 to 1 all along. If you need a map for this, what I just drew out is on like page 4 of your lab manual. I also have a video on Blackboard in the video library for this week's lab of me making this whole scheme again. For a quiz, you might get asked, I take 1/10 of a milliliter out of a 10 to the minus 2 bottle. What does that plate get labeled? Correct, 10 to the minus 3, because it's a tenth of the 10 to the minus 2. You could see questions like that. Next week, I'll show you how to calculate back based on the colony forming units. But for this week, I just want you to focus on the dilution scheme and making the plates. So for M. luteus, you're going to do a spread plate, 10 to the minus 5th and 10 to the minus 7th, and a pour plate of 10 to the minus 3rd and 10 to the minus 4th. If you're doing E. coli, I'm going to write it on the other side of the board so you don't get too confused. E. coli, your pour plate is going to be 10 to the minus 5th and 10 to the minus 6th. And your spread plate is going to be 10 to the minus 5th and 10 to the minus 7. E. coli tends to be a little more robust, so we want to dilute it a little bit more for the pour plates and the spread. So now I'm going to demo how you do this. For this procedure, each group is going to need 3 bottles of water. I've just labeled the bottles of water with the organism name and the dilution factor, and I like to set them up in order. That way I know where I'm at. I've written a bunch of pipettes, and I took per group 2 nutrient agar plates, and then you're going to need 2 empty plates as well. Those are for your pour plate method. I'll show you. You're going to label the plate as you usually do on the agar side, your initials, the organism name, the dilution level, so what dilution this is on the plate, and the date. For the pour plate, the empty Petri dish you took, you also need to label that. You're labeling the bottom of the Petri dish, so the narrower, thicker part. That's what you're going to be pouring your sample into. If you pour it into the lid is too shallow, you're going to get a big spill of agar everywhere. Always label the thing you're putting your plate, your sample into. So we label the test tube, not the lid. We label the Petri dish bottom, not the top, not the lid. You're also going to need, for the first time, your pipette pump. Blue guy in your, I'm going to do E. coli all the way through. I have a sample here, about 3 ml of E. coli culture that's very turbid. I need to firmly put that on, it might leak. We don't want that. I also open my pipettes at the top just by pushing the glass through the paper. That way it stays sterile even if I put it down on the bench because it's covered. So I put that into the pipette. I'm going to loosen the cap on the bottle I'm going into, and I'm going to loosen the cap on this plastic. It's my culture. I'm going to take out 1 ml of culture, so that's to clean 2 of the big lines here. So I'm going to fill up the pipette just to number 9 here, and that'll be 1 milliliter of culture. Open it up. Again, I don't put anything down on the table. I take 1 ml. Sometimes I like to mix it by just pipetting up and down with my thumb on the wheel like this. But I fill it up to about the 9 line with the meniscus in the proper place touching the line. If I start to see drips, that means it's not seated firmly. I can put it down, eject it, and try again, pushing it in a little further. Don't spill bacteria on the table. So put it right here and eject the bacteria into the water, and then I pipette up and down a little bit to rinse. We don't dip the entire thing into the water because that'll take bacteria from the outside of the pipette and put it in the water. So just technique. I'm then done with this tube. I don't need it again. Then I mix this gently. Remember, these caps are not watertight, so this is about as aggressive as I'm going to get mixing it. I am going to make sure it's closed though, on it open. Because this now contains bacteria and I don't want bacteria spilling everywhere. If your bottle is already labeled, like this is 10 to the 4th, just label it anyway. Label and ignore that first label. Sometimes they don't come off well. People didn't alcohol them before they put them in the autoclave. So I'm going to open my 10 to the 4th bottle and my 10 to the 2 bottle. I'm going to take 1 milliliter from the 10 to the 2 and put it in the 10 to the 4th. I'm going to take this other pipette and just seat it really firmly. When you're done, the pipettes go in the disinfectant bins at each These are glass pipettes. If you can't get it off your pipette pump, call me over. I will help. I don't want you to squeeze too hard and we don't want broken glass. So I am just going to take 1 ml out of here. And again, make sure it's not dripping. And put it in here. You can see this cap has been very over-sterilized and overheated, so it just broke apart in my hand. Put that in, and I can just take it off by bending. Okay, so this is sealed well, so I'm going to be extra careful when I shake it. Just give it a little shake. That's sufficient. For these purposes, that's sufficient. Now I loosen that cap. I loosen the one it's going into. I'm going to take my pipette, seat it firmly again, right in there. And I'm going to take 1 milliliter out of 10 to the 4th. Again, I get a little bit sometimes. That way, 1 ml out, and 1 ml in. Then, lastly, I mix that, and I'm done. Since I'm doing E. coli, I'm going to do my spread plates first. I'm going to do a 10 to the -5th and a 10 to the -7th. I've already labeled these plates. So, one is labeled 10 to the -7. I'm going to put that down in front of my 10 to the -6th, because I know that's the bottle it's going to be coming from. I put my 10 to the -5th down in front of my 10 to the -4th. And this is where you can break things up amongst your group, have one person do the 10 to the -5th, one person do the 10 to the -6th, and you're all set. So for this, I need a 1/10 of a milliliter. A 1/10 of a milliliter is going to be the distance in these two little lines. any of these two small lines is a tenth of a millimeter. Is there a 10 line? I'm turning around. Yeah. Everyone gets any two little lines will be a tenth of a milliliter. Between there. Between here and here is 1 ml. Between here and here is 0.1. So it's a really small space. 0.1, 1. Any two little lines.
0.1.
0.1, yeah. So, I open up my pipette tip. And if I do it just at the bottom of the pipette, I'm not going to get as accurate a measurement. So this time I'm going to fill up more than I need and then just eject between the two lines. If you just want to eject between any two lines, I'll show you how I do it. So, first things first, I loosen my cap, just like so. The other reason you don't want to really, really shake these is because you don't want to get bacteria on the cap, because then when you take it off and loosen it, it can germ onto your hands. So make sure this is nice and firm. And then, I'm going to suck this up. Hopefully feel comfortable for me to see. Usually around 7. And I'm going to eject from the 7th line to that first little line. So it ends up being about 3 drops. And then, since this is disinfectant, we don't mind. We're just going to put it right in the disinfectant.
Oh, it's like,
This is a spread plate. So, I like to put them on the agar first and then spread. So I'm going to do this one, which is 10 to the 7th. I need a new pipette. Use a fresh pipette every time because I don't want to carry over bacteria from the 10 to the 5th into the 10 to the 7th. I want to be as accurate as I possibly can. And I just tried to turn it. What I just did was I tried to turn it so that the numbers are facing me because I wasn't paying attention when I had it on. In reality, what I could do is just take it off and put it back on again, and that would make a lot more sense. So, just make it comfortable for you, which I did not, but lesson learned. Loosen. I should have done that before I put it on the tip as well. And suck it up to about 7. This looks a little off. There we go. And run it against the side of the bottle, just get it rid of any drips that I might have picked up. And then, so it's about 4 drops with these pipettes. And done. So now to spread these, to put the spread in spread plate, I'm going to need one of these glass spreaders. First, I'm going to seal that up so it doesn't spill. Put it to the side. Make sure your caps are always on firmly. You're done with these for now. You'll need them again for the pour plate. I sterilize the wide end, the bottom of the L of the glass spreader 10 seconds. These aren't going to get as hot as your metal loops because glass doesn't conduct heat as well as plastic, metal rather. So about 10 seconds to sterilize. And they just let it cool. Again, same rules as a loop. I'm not waving it around in the air. I'm not putting it down on the bench top.
If you don't have the glass, you have the loop.
You have a glass. Everyone has
I
a
mean, like in another environment, would
You wouldn't have that environment. If you're doing a spread plate, you would have a glass
So you
spreader.
always use it.
Or just the closable
You
spreader.
never use a loop.
You don't use a loop for this. Wrong tool. So what you do is you just touch it to a sterile area of the plate, and you can see if it sizzles, it's still too hot. Then to spread it, don't get too enthusiastic about it, but you're just going to take that droplet and spread it with the glass rod over the surface of the plate, and rotate the plate slowly between your fingers to get a nice even coat. You want to do this until the plate starts to feel just a little bit dry, because you really want to spread that bacteria around in a nice even layer. If there's a clump or droplet on there, what's going to happen is next week, all your colonies are going to be in a clump, and you won't be able to count them as efficiently. These are too close together. So the goal is just a nice even spread. This is all spread. It's nice and dry, so I can flip it over immediately. If there's still some sort of liquid on here, you can leave it agar side down until it's dried more completely. That's just a judgment call. At the end of class, you're going to flip these over, store these in your drawer over the week. So again, I just sterilized my glass spreader. And a very obvious sizzle, so I'm going to wait. Be a good person. Because if I put it in there, I'd kill some bacteria off, and then my count would be far less accurate. So this seems like a lot. I'll tell you, when I used to do this in my grad school lab, I was doing mouse experiments, and I'd have about 20 mice infected with my wild-type bacteria, 20 mice infected with a mutant I was testing. And we take tissues from them. And you have to plate the tissues to determine the number of bacteria in those tissues. I would have to plate about 4 dilutions from each tissue from each mouse. So 4 times 40. That's how many plates I would spend the whole day doing this. And then counting them was fun too. But, and you have to do it in triplicate. That's why we have people doing, we have 3 groups doing one, 3 doing another, so we can average them at the end and get a more accurate count. So I look back and I think I spent my 20s just counting bacteria on the plates. It becomes very meditative though. It becomes almost relaxing because it's so repetitive and you just zone out and just count. But yeah, it's a lot. This technique is very labor intensive when you do it for like a research or diagnostic purpose. And it takes a lot of equipment, so it can be very expensive too. So one of the learning goals for this week is name one advantage and one disadvantage of each technique. The disadvantage to this technique is it's time and labor intensive and it's expensive. So I'm just spreading it. That's why I also only have each plate.
So when you take the tissue from the mine, you have to do it by yourself, or somebody else give you the tissue and you just...
Oh no. So most people can do it on their own. So what you have to do is you have to use to sacrifice the mice by lethal injection. We used to give them ketamine. And then we would dissect them to take out the tissue. I have very small hands, and I couldn't effectively hold the mice to give them the injection well. So my PhD supervisor used to come in the lab with me to help. So he used to help me. But sometimes people would do it all on their own. Yeah. I didn't like mouse work.
I know.
And it's not something I would ever repeat again. It was necessary for that project. And we did it as little as humanly possible to do it. Most of my experiments ended up being in either tissue culture, which uses mammalian cells and culture too. Or I did a lot of microbial genetics. We used a genetic approach. So I was working with a lot of DNA cloning as well. But this particular technique stands out because it was just so time consuming. But you had to homogenize the tissues too. So you put them in a little tissue bag with some saline solution, and you have to imagine one time the bag popped. And that was also not fun. I had a mouse all over me. Memories. Okay, so now the pour plate. 10 to the minus 6. Any questions about the spread plate? Okay. So the pour plate, I have 10 to the minus 6. Why did I label both 10 to the minus 6? Oh, it's my handwriting. It should be 10 to the minus 5th and 10 to the minus 6th. This is a 6, my handwriting, and this is 10 to the minus 5th. So.
You mean this one is right?
Yeah, so these are the dilutions I'm going to, but I wrote it over there as well. So that was what I was looking at. So the 10 to the minus 6, how much of the 10 to the minus 6 dilution am I going to put on this petri dish? 1 milliliter, right. So I have my 10 to the minus 6 bottle. I'm going to put 1 milliliter here. The reason we don't plate a 10 to the minus 6 on the spread plate is because it's too much liquid. It'll slosh all around and not absorb and be messy and have a spill. So we can do a larger volume on a 4 plate. So just seat it firmly and I'm going to take 1 whole ml. Always loosen your taps before you start this, just because it makes things easier. And you're leaving around your pipette a little bit less. So the 9 line, because that's 1 full ml. And then into the bottom of the petri dish, again, remember you're putting this into the thicker, narrower part. You just put the full ml and then right into the discard. Then, what do I need for the 10 to the minus 5th dilution? What bottle am I taking from? Yep. You can look right at that handy diagram, and I expect and encourage you to. How much?
0.
Yep, 0.1 because I want the 10 to the 5th.
That's what you said between the 2 lines, right?
Yep, between the 2 little lines, and 2 little lines of your choice. Ooh, that was almost something. So I'm going up to the 8. I'm going to put about 4 drops right.
Oh, you went up to 8 because you took the 1 out.
Yeah, because it's too hard to do it between the very bottom. And I have more volume than I need, so if I waste a little by discarding it, that's okay. So now for these pour plates, what I need to do is I need to go get melted agar from the room next door. So when your group is ready for this, you just let me know. No one do anything while I'm gone. I'm just going to get to the lab next door. I'm picking up 2 tubes of molten agar.
Like, this is actually for full spray, right? Not for spray. Yeah, but I mean the circle,
Those are like the dishes. So
Okay, so I have my molten agar. Great. No one's in anything. Perfect. So all I'm going to do for this is into the bottom of the plate, I'm going to pour this warm molten agar. So it's a little hot. So you might want to let it cool just a little bit before you do it. But all I'm going to do is pour it into the plate, and I'm essentially pouring a petri dish over my sample. Now, if your agar is pretty full, you don't need to use the whole thing. So I'm just going to pour this right here. I try not to pour directly on the agar, and just until the bottom surface is covered. So if you have a little extra and the bottom surface is covered, you can stop. To mix it, we're not playing like air hockey or anything. We're just going to do it very gently, small circles, because we don't want to spill it. Small circles, just like that. Then you're going to push it to an area of the bench that you're not going to touch for about half an hour, and you're going to let it solidify.
and it's gonna be dry.
Yeah, it'll solidify. It'll solidify. The bacteria is going to grow within and on top of the agar. So you pour it, try not to pour it directly on the drop, and then gentle mixing. Don't be too enthusiastic. And then you can just push it to the side where you're not going to touch it for a half hour. And that's it. So any questions?
You're very confusing about the, like, sorry.
So the dilution scheme, if you're lost, the picture, all you have to do is follow the picture in the lab manual. Or the picture I drew on the board.
Molten agar when these 2 items.
Whatever is next to the pour plate is what you do. It's different for each organism. So if you're doing Ameludius, your pour plate is going to be a 10 to the minus third and a 10 to the minus fourth. If you're doing E. coli, you're going to do a pour plate of 10 to the minus fifth and 10 to the minus sixth. So for your next lab quiz, which is not next week, you need to know this
I
dilution
understand
scheme,
about that.
know how to make the dilutions, and know how to plate them. So get your bottles for groups that I split you up into. Here are the cultures right one for your group? Yeah?
If you take out one,
Do it just the whole row because she was late and missed part of
say
it.
there's like 1,000 bacteria in this one, right? If you take out 1 milliliter, then you can expect there to be 10 bacteria in that 1 milliliter, right? Because you're expecting that the bacteria are evenly distributed. So 1 milliliter will have 10 bacteria if there's 1,000 bacteria in it, right?
And it's like, sorry, in that, how many in 100
If
milliliters?
there's 1,000 bacteria
1,000.
and in 100 milliliters, and you take out 1 milliliter, then you expect that you're going to have,
But you'd only
yeah,
have
you're going to expect that you're going to have 10 bacteria in that, right? But if you take out
Yes, if you
0.1
subtract.
milliliter, you expect that
So
you're
each
only
group
going to get
gets 2
1
nutrient
bacteria,
augars
right?
and 2
You're gonna get
empty plates.
1 bacteria.
So this is how,
Yeah, that's how much of the solution you take out.
how much one that you draw.
If you took out 0.1 milliliter, you expect to get 1 bacteria. If
Yeah.
you have 1,000 bacteria in 100 milliliters, right? So you expect to only get 1 bacteria in this, but you get
You're
10
jumping
bacteria.
ahead to next week.
Yeah, but I'm saying, like, this is why you
Yeah,
label it
this
with
is-
different numbers.
yeah, it is.
Because,
Yeah. For today.
well, we're working backwards from this, right?
Right.
We're working backwards. We're only going to get this number ultimately. But this is why
If
the
that's
logic.
their how-
this
how-
is the logic of it.
Yes, that is correct. You're just jumping ahead to next week. So for today, just focus on the physical process of making these solutions. And then, so what organism are you doing?
We're doing.
Gluteus? Okay. So you're going to make these 3 solutions first. So 1 ml, 1 ml, 1 ml. Then for Gluteus, spread plate, I'm going to take 0.1 ml out -4 for the 10 to the -5, and 0.1 ml up to 10 to the -6 for your 10 to the -7. So those are your spread plates. For your pour plates, you're going to use 10 to the -3 or 10 to the -6. Draw it here at 0.1, makes that 10 to the -3, and 1 ml of your 10 to the -4 for your pour plates.
All similar to this, gonna be 9 and 2 and 3 at the same time, we only do one.
Well, yeah, but you need to make all 3 bottles.
Yeah.
And, oh, I forgot to tell you, when you're done with your street plates and you've looked at them and you've recorded them, you can put them into this card and we're not doing them.
A stand? Use my stand.
Never label the lid, always label the thing your sample looks into. Yep, the thicker, narrower part is more than What is the name of the You're doing microscopic studies, so you have to write that on the plate. Your 10 to the -2, we're not doing a 10 to the -2 solution of microscopic studies. You have to make your solutions first. For your 10 to the -3 pour plate, that would be a spread plate with agar. You're doing a 10 to the -3. For the spread plate, you're doing a 10 to the -5 and a 10 to the -7.
The other.
Yeah, that's a spread plate. The empty petri dishes are your pour plates.
This one is this.
They don't fit in there. Someone needs to hold it.
Oh, okay, that's.
Or alternative.
So you take a bottle.
That's it. What's wrong with those? They have condensation. These?
Yeah.
Over there.
There you go.
That's for the spread plate. So the agar, like I just demonstrated, the agar that's already in the plate is for your spread plate. The who has not used their bottles yet that is doing Amludius. Oh wait, let me see if I can just get for you. We'll get you. If you need something to put that big plastic tube in, I have little holders up here you can borrow. So these little holders up here, just return them when you're done. Thank you. Okay, here you go. Sorry about that. Count it.
This one should also have the
Oh, yeah. You're doing what organism? Amludius, so your spread plate is going to be 10 to the minus 5 and 10 to the minus 7. And your pour plate is going to be 10 to the minus 3 and 10 to the minus 4. Yeah, we'll be right there. What's up? Do your best. One got a little shorted. You wanted to have a impact? And just put a note in your notes that that bottle was a little short. That way if your results look a little funny next week, you know why. And remember, the pipettes go directly into the disinfection.
Yeah. Start by keeping
Yeah, when you're ready to spread, you do the glass
Would it be
bottle.
easier
When you're
if it,
done
like,
with
try to put it
your
through the paper
culture,
thin?
you can put it back up here.
You got it.
Nope, the cotton stays in there. The cotton is designed to keep from bacteria from getting into the pipette. It's a safety feature.
Did you, like,
Yes, they did. How much is on one?
yeah, 1 milliliter from
What's that? You need the glass spreader? I'll leave them right over here. Glass spreaders are right over here. You need them. Just remember to sterilize it. When your pour plates are ready, just let me know and I'll go get you your agar.
we're gonna do
0.1.
yes, we're gonna have this one, we're gonna take 0.1 of this one.
So this way you show me my inputs and then you spread it.
All right.
You don't have agar yet. You didn't ask me to get it. Oh, you just wait until it
0.1.
dries a little. Yeah, you could leave it like that for like 10. Sorry, I misunderstood what you were asking. I was like, I didn't give any agar yet. So that's already solidified. So the second this surface seam straw, you can flip it over. You need the agar? Okay. Shredder's right there.
That's different one.
The ultimate.
And staples. So this is not glass. It is what it is.
These shots, right?
0.1 ml. It works out to about 3 to 4 drops for 0.1 ml. Okay. Does anyone else need agar for their pour plates at this time?
I didn't take it yet.
What was that?
We're still doing procedures.
Okay, that's not what I asked. I just asked, does anyone need agar right now? Okay.
Point one of this.
So this time I'm gonna do a drop like the other one, or I just gonna take 0.1.
It's the same as this one. You want to take,
Same.
yeah, just between the distance between two of the little lines.
It's a little warm. Who asked me for it? It's a little warm. This one's a little fuzzy. So you make your glue.
Tenth of 0.1. Yeah, 0.1 of one whole.
Right, okay, let me check it out, and, I will get back to you, and I'll let Alicia know. Because, yeah, they need to be able to work.
Right, so I told them I'm gonna go in. Whoever is wearing these kind of glasses here now, so who's wearing so 1, 2. She bought
So this one needs the same amount as these, just
she has ones that fit over her
between the
glasses.
two. On this one.
Well, I can give you what I have, I can get some.
I think it needs to go from one small line to another.
There's only one small glass.
Louise? Nope, she's good. I just need to see your glasses.
There's only one
5,
small
6,
size.
in this
Yeah?
batch.
Yeah.
All right.
And then in my other section.
Okay,
Today we're doing 12:30. Thank you so much. I was very confused. Yes, I'm gonna go get the hour in a second. I just have to wash my hands and I'll go back home. Who needs that right now? 1, 2, 3. Okay, so I need to know how many I need to go get.
Oh.
6,
We need
8, 9, 10. Okay, everyone.
Only one line.
Let me put my goggles back on. Sorry,
That's
that was
for, yeah,
our
that's for this
environmental
one.
health
And for
and
this one, we need a whole milliliter.
One milliliter.
So yeah, unfortunately, the kinds I prefer is no longer okay. And that just changed last week, so I apologize to walk hours for this class that are no longer okay. I am in the same boat as you. I just bought for emergency 20 packs of those type of goggles, and the students forgot them, and now I have to return them back. Okay, 10 to the hour coming up.
Like one of these, not like a line between.
We'll see.
Yeah, that's. Yeah, the middle.
Okay, we need to take 2 shoots. They're a little warm. So actually, you know what I'm going to do? I'm going to leave them up here. In about 1 minute, when they cool a little more, come up and take your 2 tubes. So count to 60 and then come on up. racks. Or if you have a partner, your partner does one, you do one, you're all set. Then just leave them on your bench, just solidify and remove on what's selected and different.
No, I think it'll be.
No, they withstand that. It's fine. It's only about 55 to 60 degrees Celsius, which they all will survive. They don't like it, but they'll survive it.
Oh, there's a little
That's
something
fine.
in the bottom.
That's sterile, so. Thank you for letting me know. No, that's the absolute right thing to do. I'm just reassuring you that if it's sterile, it doesn't count as like a spill we need to decontaminate. But thank you. Thank you for your esteemed tip. Okay, when you're done, what I need you to do is take a little alcohol on a paper towel, remove the labels from your dilution bottles, and just put all your dilution bottles over here, nice and tightly capped. Yeah, remove the labels from the bottle. Remember to return your big plastic tube up front. And do it before I'm missing one Amelia. And there's the E. coli. You beat me to it. And take a moment, wash your hands. Wash your bottles off with the alcohol. And just put them next to that metal basket, just next to it, not in it, next to it. Because that's too full already. Thank you. But remove the labels. Yeah, those just gently, carefully. Just splash with this. All right, over there. Don't put them in the basket, please, just next to the basket. Thank you. Turn your glass spreaders as well as Okay. So the other major topic of week 3's lecture we also cover in lab. Next we're going to be talking about optimal, minimum, and maximum growth conditions like temperature, pH, osmotic pressure. But today we're going to talk about selective and differential media. So, media is the thing we use to grow bacteria on. You all just inoculated bacteria into a media that is not selective and not differential, meaning a selective media is one that only certain species will grow on, based on some sort of metabolic characteristic. The media you've been growing on so far is more of a general growth media, meaning everything should grow on it. It's not selective. So selective media, and this is all on page 8 of your lab manual. Only certain species grow. It's like a dance club with a velvet rope. Only certain people get in. You have to be dressed right. You have to have a certain metabolic property. So only certain species grow on a selective media. The other type of media we're going to look at today is differential. It's like a differential stain, except now it's a media. A differential stain allows you to tell some difference between a bacteria. A differential media makes the bacteria appear differently on the media depending on some metabolic property. It might turn a different color, or the whole media might turn a different color based on something it can do. So differential media allows you to tell the difference or differentiate between species based on their metabolism. We're going to look at 4 different types of specialized media today. I'll go through them one by one. Some of them are selective and differential. Some of them are only differential. So the first agar we'll talk about is blood agar. Blood agar is going to test for the properties of molecules. So it's differential or hemolysis. Hemo, blood, lysis, bursting red blood cells. And to test this, we use a type of media called blood agar. For each of these media, I want you to kind of notice what they look like before you put anything on them. So blood agar is red because it has sheep's blood in it, defibrinated sheep's blood. All this is TSA, so tryptic soy agar, TSA, plus 5% sheep's blood, SB. It says so right on the plate. Now, when you take this plate, we're going to notice that I can't see through it. It's opaque. It's not see-through because the red blood cells block the light from reaching my eye. So I can't see through it. It's opaque. We're going to grow different species of bacteria on this media. This media is not selective. All the species will grow on this media. But we'll be able to tell the difference between species based on their ability to burst red blood cells. So, something can be what we call alpha hemolytic, the Greek letter alpha. Alpha hemolytic means partial bursting of red blood cells. And if it's alpha hemolytic, it's going to make the plate turn a greenish color. Kind of like if you left a Band-Aid on a cut too long, it starts to turn that like brownish green color.
That's because of
Not the lytic cycle, that's something viral. This is because of the damage to the red blood cells. So the bacteria produces something that damages the red blood cells. If a bacteria is alpha hemolytic, it only partially damages the red blood cells and causes a greenish color to develop in the agar. If something is beta hemolytic, it means it completely bursts the red blood cells. So complete lysis. You'll be able to see through the plate because the blood will clear. You'll hold it up to the light and all of a sudden you'll be able to see behind it. Because the bacteria have destroyed the red blood cells completely. That's beta hemolytic. So a lot of times you hear beta hemolytic streptococci. That's the bacteria that causes strep pyogenes. Gamma hemolytic means it has no effect on red blood cells. So the plate will pretty much stay unchanged, and you'll just see the bacteria grow on the plate. Again, these are all reviewed in the next section over from the serial solutions in your lab manual. Goes into each one, what alpha, beta, or gamma is. That's the first type of agar. The second type of agar we'll use is something called mannitol salt agar, or MSA, just the main word. MSA is mannitol salt agar. It is also red, this kind of bright red. But different from the blood agar, I can see through it. So MSA is both selective and differential. So only certain species will grow, and we'll be able to tell the difference between those species based on what color they turn the plate. This media is selective because it contains 7.5% sodium chloride, or salt. Salt will inhibit the growth of certain organisms. This will only allow the growth of what we call halotolerant organisms. So halotolerant organisms they don't need salt to grow, but if it's present up to like say 10%, it doesn't care. It can grow anyway. It tolerates the presence of salt. If something is not halotolerant, that much salt will prevent its growth entirely. So that's how it's selective. Only halotolerant species will grow because they're 7.5% salt. What makes it differential is it contains a special sugar source, special carbon source, called mannitol. And we are testing the ability to ferment mannitol. Fermentation, as we'll learn in week 4, is a process by which bacteria can make ATP. Small amounts of ATP. And a byproduct of fermentation, in this case, is going to be acid. What does acid do to the pH of a solution? It lowers it, right? The pH will drop. We're going to detect that drop in pH with a chemical called phenol red. Phenol red is red at pH 7. pH 7, it's red. However, if the bacteria is able to ferment mannitol, that's going to drop the pH, and at pH 6.8, mannitol red turns yellow. It's a very sensitive indicator of pH. Mannitol. So mannitol is the carbon source. Phenol red is the thing detecting the pH change. It's not fermenting the mannitol red, it's not fermenting the phenol red, it's fermenting the mannitol. The phenol red is what's detecting the change in pH. So if the bacteria is halotolerant, it will grow on this media. If it doesn't grow on this media, it means it's not halotolerant. It's differential if it grows and the plate turns red, or stays red, however. That means it's mannitol negative. It does not ferment mannitol. If it turns yellow, that means it's halotolerant and mannitol positive. Take for me when she said they'll be in by next week.
Okay.
So if you just want to leave those,
Sorry.
because we're going to set up an experiment. So if it doesn't grow on this media, that means it's not halotolerant. If something isn't growing, it's not going to make ATP. So it's not going to ferment anything because it's not growing. So if it does, thank you so much. You're welcome. So if it doesn't grow on MSA, it's inconclusive. You can't say whether it ferments mannitol or not. You can't tell anything else about the bacteria except that it's not halotolerant. Okay, this becomes important later on because you can't assume it's mannitol negative if it didn't grow. Because under different conditions where it does grow, it might ferment mannitol. So from this, if it doesn't grow, all you can tell is it's not halotolerant. You stop there. If it grows, you're going to look to see if the plate is red. If it's red, that's mannitol negative. It does not ferment mannitol. If it's yellow, it means it does. Okay? So that's the second type of media.
Inconclusive was if it doesn't grow on
MSA.
MSC.
If it doesn't grow, you don't know. It rhymes. You can remember it that way. If it doesn't grow, you don't know if it ferments mannitol. So the third type and fourth type of media I'm going to do together because they test for pretty much the same thing. We have MacConkey agar, which is a more purpley red see-through media. And we have EMB, or eosin methylene blue agar, which is also see-through, but a much darker purple. So, these media are both. So this is MacConkey. And then we're going to talk about EMB. MacConkey is both selective and differential. EMB is both selective and differential, which means, again, not everything will grow. And what does grow, you'll be able to tell the difference by how they appear on the agar. So MacConkey agar is selective for gram-negative organisms. And I always think this is so cool. You don't have to do a gram stain. You can just plate it on MacConkey agar, and if it grows, then it's gram-negative. It's selective for gram-negative because it contains two things: crystal violet and something called bile salts. So these two things inhibit the growth of gram-positive organisms. So gram-positives will not grow on this media. And we know from our gram stain, gram-positives love to absorb crystal violet, so that's part of what's killing them. EMB stands for eosin methylene blue. And this is a dye that also inhibits gram-positive. So it's also selective for gram-negatives. Gram-negatives will grow. So if it grows on this agar, it's gram-negative. If it doesn't grow, it's either gram-positive, acid-fast, or mycoplasma. But it's not gram-negative. Okay? It's differential for lactose fermentation, both of them. Because it contains lactose. So we're testing for lactose fermentation. If lactose is fermented, the pH drops. On MacConkey agar, there's a pH indicator called neutral red, which turns colonies purple if acid is present. So purple colonies on that plate means lactose is fermented. On EMB, it also contains lactose, but it doesn't have any additional indicator because under acidic conditions, the EMB dye will precipitate or fall out of solution and create a metallic green color. So, if it doesn't grow, you know it's not gram-negative, and that's it. But you don't know if it ferments lactose or not, so you would say inconclusive for lactose fermentation. Because again, if it doesn't grow, you don't know about fermentation. On MacConkey, if it grows, it's gram-negative, and if you get purple colonies, it's positive for lactose fermentation. If you get white colonies, it's negative for lactose fermentation. On EMB, if the colonies are white or pink, it's negative for lactose fermentation. If they're metallic green, it's positive. Yeah.
So they both test for lactose fermentation?
They both test for gram-negative and lactose fermentation, yes. We call gram-negative bacteria that ferment lactose coliforms. These will test for coliforms. Coliform bacteria are gram-negative and lactose positive. And they come and live in the intestines of animals. So testing for coliforms is a stand-in for testing for fecal contamination of food products and water.
Would it like ever make sense to do both of these, or would that be completely
No.
different? Like, are they?
We do both of them because we have both of them. But in like a lab, you would do one or the other because they test for the same thing. So one doesn't add anything additional. It's a good question though, because I get it all the time. Why do we do that? They do the same thing because we have them and we pay for them and we might as well use them. So you're going to work in pairs on this. Everyone's going to get one organism. And then we have a mixed culture. I'm not going to tell you what's in the mixed culture today. I'll tell you what's in the mixed culture next week. But it's a mixture of 3 of these organisms. We'll see how the results come out. So, we're short on time. So maybe we'll do this by rows instead. We have 6 organisms though. So I think we have to do it in pairs. Oh, we have to do it in pairs. Okay. Sorry. It will be. So. Staphylococcus epidermidis. Staphylococcus aureus. E. coli. E. subtilis. Pseudomonas aeruginosa. Proteus vulgaris. Oh. How'd that happen? We were in, 3 rows. Yeah, that makes sense. Okay. I'm trying to think how we need to do this up. cotton swab. And for everything except the mixed culture. For the mixed culture, you're gonna do a quadrant streak. Because we wanna separate those colonies to see the difference. But every other plate, you're gonna take a cotton swab, and the swab to one plate. And then the cotton swab goes into disinfectant. That way we speed things up, we're not waiting for the loop to cool. Okay? So quadrant streak for the mixed culture, squiggle for everything else. Okay? And you're doing each type of plate. So you're gonna take 2 plates of each colony, one for the test, one for the individual colony, and one for the mixed culture. Does that make sense? Okay, let's go. At about 10 minutes, I might pull one person in the row and start doing a grab. Just so we don't run out of time. This lab is particularly jam-packed. If you need a refresher on the quadrant, please, I will need to know. It's not a little easier, but it does the same thing. Cotton swab program is going to get more. Remember to label what type of media it is as well, because you won't remember next to the plate might be a different color.
One for each.
Per group, not for your
Labels.
but for
I
your
see,
group.
yeah, got it.
I am going to demo a quadrant streak, but I'm going to use a 3-streak method. Okay, so if you need to see a colony purification using a slightly different method, I'm going to demo it now. You can come up. If you are one of the people that has safety glasses instead of the goggle,
You can take the note over there.
our environmental health and safety person left some over here for you. You can take a pair.
You're gonna mix controls.
Thank you.
NOS? Yeah, that's right.
Okay,
And then
so I'm going to do a
And then you mix
3-streak
both.
method.
aid.
Sterilize my loop. Oh, you don't use them anymore. No, what would you use it for? That's solidified agar. I labeled my plate. I'm letting my loop cool. So what I'm going to do is I'm going to do 1 streak with my culture. That's the first streak. Then I'm going to sterilize my loop and pull down and make a second streak right below it. And then I'm going to pull down after sterilizing the loop all the way down the plate. That's going to be my third streak. It's the same thing as the bottom, which is 1 less streak. So I've let this cool. I'm going to take these stuff on there. feather tickling the surface.
We're gonna do 4, right?
Nope, I'm just gonna do one.
Oh, 1.
I'm gonna do a 3 streak, so I'm gonna follow the scheme I put on the board. Right there, sterilize my loop. I'm not gonna go around the plate, instead of going around the plate, I'm just gonna go vertically.
So for this portion, we're doing after each.
Yes, yes, so 4 plates. So I sterilize my loop, I'm gonna let it cool.
With the cotton swab.
No, this you're doing with the loop. Everything else you do with the cotton swab on the collar. I did.
No, the.
Oh, the cotton swab you don't sterilize. It's already sterile, and then you just put it into the disinfectant when you're done. So for my second streak, what I'm gonna do is I can see the scratches on the plate where I put the first streak. I'm just gonna pull down once or twice, 3 times from the first streak. So it makes like a little tail, and then I'm gonna go back and forth to make the second line. Yeah. 1, 2, sterilize again. Now I'm ready for my third streak. The benefit of this is it's just a little faster because you're doing one less sterilization step. It works just as well. In fact, personally, I like this one better because if you get really good at it, you can fit multiple strains on the same plate, up to 8, using like a toothpick, not a loop. So once you get really advanced at that, it saves a lot of research money because you're using fewer plates. Each single plate of like regular nutrient agar is about $100.
For mixed food.
This is for the mixed color. For everything else, you're doing just the cotton swab. So I pull down 3 or 4 times, and then I go across. And now I'm going to go all the way down the rest of the loop. And that's it. And you're done. Yep. And you should get single colonies that way.
Which one do you want to do? One long?
I feel like you got a better look at what she was doing.
So you have a week.
Okay, so I'm gonna do
So
the
when
mixed
you're done with
culture.
your mixed
Yeah.
culture, please pass it on to someone else.
Bring over my table. Yeah. Like this one.
I'm just gonna move these goggles out of the way, but we still have a few more pairs if anyone needs them, if not, I'll take them from you. No, we're not doing all of the ones listed there. We don't do PA on there. We're only doing those 4. For each plate you need 1, so each group needs 4 cotton swabs. So 2 packs. Not gonna be as efficient.
Oh, this one is,
Contaminated? Yeah, if you see something on the plate already, don't use it. I have it up in my microscope. It looks pure. It looks very gram-positive stuff. So I don't know why it's wrong. Does that have filter on it? It goes directly into the disinfectant. Right there. Cotton swabs go right to the disinfectant. I know. As well, you should. I don't know. We'll see what it looks like after. I'm looking at the indicators and what happens, and then I'm gonna put it. What I said to my class study is like, let's put it in the indicator and see how it goes. Okay, that's fine. No contamination.
Oh, I'm so sorry.
Okay. The mixed culture? No, the mixed culture goes on second step. So what is the culture? Yeah.
Yeah, I was doing the cotton
Okay, so get the cotton swabs quicker. So if you are done, I need you over by the first row. You will share this information with the rest of your row. So if you were doing the cotton swabs and you're done, you have something else to do. Come on over. Have one person from every row, except, yeah. If there's more than one person, you can hold on to. Whoever's done. Are you done? Okay.
Yeah.
So if you,
still need
okay,
to
that's fine, that's fine. As long as someone's working and someone comes over here, we're good. But, so find your group members if there's more than one of you here and pair up. So this is the report template for the Nebraska. Everyone, that one already stand with your group member because they're going to need your help. No problem. Don't worry if you're busy working or we're doing this for you and your group member. Everything you're doing is for you. You're not really. You just need help doing them because I can't make 4 monographs with them. So, what we're going to do today is the experimental setup. If you're in group 1, which is furthest group left, that's you two, you are going to add no supplement to your Winograd column. I'll walk you through what that means. If you are in group 2, raise your hand. Okay. You are going to only add carbon, which means the only thing you're adding to your enriched mud is a little bit of shredded paper, 5 grams of calcium carbonate. Group 3, you are going to add sulfur. So what you're going to do is only add 5 grams of magnesium sulfate to your soils. If you are in group 4, you are going to add everything. So both carbon supplement and magnesium supplementation to your enriched mud. So the way we're going to start, and I'm going to start this off for group 4 because they're getting all the supplements. So that way everyone will see everything. So what you're going to do to start is we have spoons, but you can also just pour it. Try not to get dirt on the counter because we only have a set amount. This is soil from the Kingsborough farm. You can kind of massage it like a bagpipe and it goes right. Don't worry, we're not following aseptic technique here because this is dirt. So what I can do is scrape some of this up and use it anyway. So you want to fill the beaker, and I'll of course clean this up later. You want to fill the beaker about halfway, just like that. do encourage everyone to wear gloves for because it is dirt. There's some gloves on my. Please, the person here. It has a lot of microorganisms like that. I don't know exactly what they are. There are a lot of wild cats and geese around campus, so let's just. Sprinkling with Parmesan cheese. Put some shredded newspaper. That's your carbon supplementation. So if you're doing carbon supplementation, nice healthy handful of paper, 5 grams of calcium carbonate right in here. Then you're going to stir it up. At this point, you can take everything back to your bench. After you add your calcium supplementation, if you're in group 4, you're also going to add magnesium supplementation. If you're in the magnesium group only, you're just going to add the magnesium. If you're in the carbon group only, you're just going to add carbon. And if you're in group 1, you got off easy, you add nothing. You actually don't even need to make the enriched mud. So, then, come down here, you find your groups with a Gradsky column. I already labeled them for you. You pour the enriched mud in first, and then layer on top the other mud. Group 4, you might want to make this a little thicker before you do. You're going to tap, tap, tap. You want it to be about this high with dirt, that high with water. So you might have to add a little extra water in. But what's most important is that you leave space for the air. Because if you look at the columns that are here now, this is what you want to see. Like this one, I could probably add more water to. But you see a layer of dirt, which is going to break into other layers by color, like I talked to you about before. A layer of water, and then air. And then you're going to take some saran wrap and a rubber band and seal it up, and you're going to put it in order. So 1, 2, 3, 4 here. And then you're going to take your first pictures. You're going to take a picture of these 4 columns, front and back. Those are your reference columns. And then when the whole class is done, you're going to take a picture of these 4. Now it's going to get crowded over here. So what I'd like you to do is when you can, when you have your mud all supplemented, take it back to your bench and make the column there just so you get out of everyone else's way. And again, that moves quick, so, you know, be careful. So we're going to add a little bit more dirt to this. You can always add more dirt. You're not really going to mess everything up. I just want to show you the correct consistency. It's all going together in the same column, so it doesn't matter that quarter of it is here than the other side. Yeah, that's more like it. Thick milkshake. When McDonald's ice cream machine is not broken, what you get. Okay, and there are spoons over here. If you didn't get a report template, I will give you one that is not covered in dirt. Okay, any questions? Have fun playing in the mud. Get some gloves. Wear your goggles. Get started.
When it Gradsky column.
That's my dirty one. Okay, did I miss anyone? I have more dirt in the fridge, so. This is Kingsborough Community College native farm soil. I went on Monday to dig it up with my own 2 little hands.
Aww.
You don't add anything, you just make a column.
What about the water?
You add the water, yeah.
So we add the water, you don't have to column
You don't, yeah, you make the thick milkshake and then you're just gonna fill it all the way up into your column. Okay, let me get a container for slimy. Anyone here watch Sesame Street? Oscar the Grouch's little worm friend that's slimy?
We're on Parkland Road.
That's how you know it's good soil. Things live in it. Yeah? What's up? Another one? Now you're looking too carefully. What's up? Oh, I thought you needed something. Oh, fourth grade science. Jumping in one of your house plants. I'm in the beaker and then we don't think I was done right. Oh, like a quarter. Oh yeah, so the algae didn't come in time,
You're gonna
so we
leave
wrote
this
part
space?
about adding algae.
No, that's, she said that you wanted to be up that full with water and then the rest with air. So this full with dirt, this full with water.
Water,
that full.
air.
There's
Any of
probably
these
some
are-
algae
will we
in
start-
there already,
Yes,
like
they started
in
doing
other
it.
So like,
Again, try not to work over your notes.
It is not enough. A really upward angle.
Because if you get something on your shirt, it won't be too shirt for you.
but this thing is like thicker than this one.
So yeah, quarter of the mud goes in there. That's a little thick, so you might need a little more water in there. Or a little, like volume wise, you want it about halfway up the side, so that's gonna be about.
I don't know if anyone else wants to do this part. I'm not
600,
trying to
650 with dirt, and then you add
take over
your,
and do it, you know, laterally.
your water. That is seawater. The reason we're not using tap water is because New York City tap water has a low level of chlorine in it, which might kill off some of the microbes. That's why it's there. And when you're done doing your street cleats, you can turn off your sterilizer and unplug it. You won't be needing it anymore.
It's an awful sound. Yeah, I'm just trying to get all of it.
So,
I guess tap it so that it's flat. We're gonna add this one on top.
Yeah. Continue next one. Like, is it the same?
It's a little, this is a little more water.
Yeah. That's how she told us to do it. One is more thicker and one is more water.
street, sterilize. Come down, street, sterilize. Pull down, street, sterilize. Where's your last name?
Oh, I dropped it on your hand. Sorry.
That's this one. You need last piece.
Yeah,
Yeah, so you can see the line.
much easier.
This is today. We need last.
It's just a little bit too big to really hold with one hand.
Okay, it's enough.
Well, we want it.
Want a little bit more?
I think a little bit more because some of that is.
Okay, you're good.
Yeah.
You're good.
Let's see. If you tap it now, will it separate this?
Yeah, it's more.
Well, we want the water to separate out to
So I
the top.
suggest you get a vinyl.
Sterilized food. Let it cool. And then,
Well, every time I
yeah. But I struggle with myself.
Okay, if you're done with your winter graduate column, remember, you're going to sit on that tray. That third tray there that's empty. For your plates, they should be ready to flip now. They should be ready to flip. No, that's good, because remember, it's gonna settle down, yeah.
No, she said not to add more. I think we should wait for it to separate. Yeah, we want it to separate so that there's a layer of water on top.
So
And
what you can do, yeah, the reason I said what you can do. Pour it in.
It's all
Okay, you're all right? Okay.
do y'all have? We should make
Yeah.
it a WhatsApp.
You should put group 4. Right?
Okay, so are you done, Nubia? Group and making the address.
And I have the phone number again.
I'm just gonna collect cultures if I see them that are mature.
Are we gonna do WhatsApp or
Can you put in your phone number here?
Yeah,
Because
make sure
only
you change
if I have your
the
number in a contact, I can be able to add you in a WhatsApp.
Oh, I thought we can do like QR
I'm
code.
missing one staff at service.
Yeah, I mean whatever.
Google Drive? Or
Yeah, let's do a Google Drive.
that's good. We'll make.
I was saying that we can just do a QR code for WhatsApp.
So you want to create it?
Smart. Yeah. I can set up a.
Because I think this is like quicker. Like if you can do it quick, it's fine. Did you did it? Okay.
Wait, let me see if we can do group.
Where's the L?
That's my name.
I see.
Oh no, I can't create group first. I have to. Let's see if it works.
Okay, gotcha. Yeah. What's your last name?
Yan.
Yeah. I add you to the group chat. Do you see me or no?
I didn't get the request yet. Let me see.
I don't see anything, so that's why. But I just scanned the code and then I don't know if that's a group or only your phone number, so that's why.
No, it's just my phone number.
So that girl,
If not a group, we can create a group first.
okay. I just gonna create that anyway.
I am missing one mixed culture
Okay,
and
I
one
got her.
Staphylococcus epidermidis.
I create a group.
That was the back two and over here.
I don't have your number. Let me.
When you send me a message,
You can add her in, right?
Yeah, so leave the beakers
you
all
should add me.
have them.
I already add all
Just make
you.
sure that
I already
they're
add you.
I know, but
And then,
you have everyone?
if
I only add
someone's
her,
white
but
down,
that's
then
why.
they're
Can you do for her?
white down.
Like, do you have her number?
So that's why you have to message me so that I can
Very
add you,
important for your
then I can
attention.
add
I am
everyone in
missing
the
one
group.
Staphylococcus epidermidis and one mixed culture.
I already
We
like
cannot
created
leave
a
until
group
those
after
are
you telling
accounted
me
for.
that you. I only have your number, so that's why I created.
That's why everyone gets mine. I create
Please
the
help
group.
me find them.
I created another group, so you don't have to create another group. Just you this group, okay? And then if you don't have her number, I just gonna add her.
I'm just missing a staph epidermidis.
You have my number, right?
I think so.
Yeah.
And you should be able to find her in WhatsApp.
I mean, it's easy, right? If not, just put your number here and save yours. Okay, I got everyone.
But you didn't invite.
Okay, so you're using staph epidermidis. I'm missing staph epidermidis. Okay, you only took one?
Yes, that's the problem. Because you turn off.
I'm missing a staph epidermidis and your group did staph.
I already give it back to you.
Okay, we're
So
still missing one though.
I just gave it back to you so I know that for sure. So I think your WhatsApp is like set up as a private, so I cannot like invite you to join my group. You have to like unlock your private setting.
I'm in other people's groups.
I know it's happened to me with another person as well. So because I don't have your phone number in my contact list, that's why only on
That was the
WhatsApp.
QR code.
I think QR code only for WhatsApp contact list.
No, my number was on the bottom.
Yeah, it is, because I have
Let me redo it again.
Everything's good. Make sure when you put cultures back, you put them in
Yes.
the row they belong. Otherwise, they're only counted for. All right. So your Winograd people, you keep them.
Okay, now I can add you in.
Yeah, because you didn't message me for me to add you.
No, don't store eyewear
both
in your
message
drawer
me.
because
So I have to message you like privately.
No, first you scan it so that you get my info, but then you didn't request the ad.
All right.
I save your number and then I
So-
add from this group,
Too
but
bad, not too messy.
but now I can. So the problem solved. Did you see your invitation?
you already added it?
Let me redo it again.
Okay, so we're ready to clean up. It's 12:24. Make sure you put your plates in the drawer. We're missing one Winograd column over there. Yeah, we can seal that. Okay. a lot of calcium carbonate over there. Please get a paper towel and wipe it up. Yeah.
Cover your nose.
What's up? Oh, okay, great. Thank you. Thank you. Oh. Okay, well that's teamwork then. Okay, get rid of that and then wipe down. Get like Thank you.
Yeah, well, it looks like it has a little bit, but
It will. It'll stay or it'll settle out. Trust the process. It'll settle out.
Professor, where we're gonna
Okay, so the dirty beakers just leave near the others at the end of the bench. Thank you so much. This class is very labor intensive and there's a lot to get done. I'm sorry if things are very fast paced today. It doesn't happen anymore, but it does happen. I appreciate your flexibility and attention. Okay, DWNZ. What is this? Why is this left up as sterile? I don't know either. And then there's, okay, that was in your report. What about this one? Label it and keep it in your report. Don't leave it back for the sterile stuff, because then it's gonna get mixed in and someone else might get it. Not that it's unprofessional, it just leads to mistakes. We wanna try and minimize that, so.
Alright,
It happens, we're all learning here. No one's an expert yet, not easy.
She said that she wanna add us to the Google Drive as well.
yeah, I
Make
was,
sure your legs are unplugged.
so. If,
Make sure you tuck in your chairs before you leave. My legs and toes will thank you.
Kim, can you put in
And I will have those
when you're done?
graded this afternoon.
My first name and last
Remember, the top
Put in your-
score is a 50, so if you get a 50 out of 50,